Welcome to Human Physiology. Today, we are diving into hormonal regulation, the incredible biological cruise control system that keeps your body perfectly balanced. We will explore how your body knows exactly when to release hormones and when to stop, ensuring your survival and health every single day. Whether you are completely new to this topic or looking to deepen your understanding, this lesson will build your knowledge from the ground up. To understand how this works, imagine you are driving a car on a highway and want to stay exactly at 60 miles per hour. If you go too fast, you ease off the gas. If you go too slow, you press down. Your body does the exact same thing with its internal chemicals, called hormones. Hormonal regulation is the precise physiological mechanism that controls the synthesis, secretion, and degradation of hormones to maintain internal homeostasis. Without this, our bodies would rapidly spiral into fatal states of imbalance, like severe dehydration or hypoglycemic shock. Now that we understand the goal is balance, let us look at how the body actually achieves it. This process is primarily governed by negative feedback loops. The endocrine system relies on the hypothalamus and pituitary gland to act as central command centers. They constantly monitor the blood concentrations of various substances. When a specific hormone reaches its optimal level, it binds to receptors on the very glands that stimulated its release. This effectively shuts down further secretion, ensuring that target cells receive exactly the right amount of chemical signaling. Let us visualize this command chain to make it completely clear. On screen, you are seeing a schematic of a negative feedback loop. At the top, we have the hypothalamus, which sends positive signals to the pituitary gland. The pituitary then stimulates the target gland, such as the thyroid, to release hormones to the target cells. But notice the red arrows looping back up. Once the hormone levels are sufficient, they send an inhibitory signal back to the hypothalamus and pituitary, acting as a brake to stop further production. Having seen the biological pathway, we can now translate this process into mathematics. To truly understand how hormone levels are maintained, we must look at the relationship between how fast a hormone is produced and how fast it is destroyed. The concentration of a hormone in your bloodstream depends on a delicate balance between its secretion rate and its clearance rate. By modeling this relationship, we can predict exactly how the body will respond to changes in gland activity. Pharmacologists use this exact mathematical approach when designing artificial hormones. This balance between production and destruction gives us our first key equation. The formula states that the rate of change of hormone concentration over time, denoted as dC over dt, equals the secretion rate, S, minus the clearance rate constant, K, times the current concentration, C. Conceptually, this means the hormone level rises based on what the gland pumps in and falls based on what the liver and kidneys filter out. If the secretion rate increases, the concentration rises, which in turn makes the clearance term larger until they perfectly balance each other out. Before we move on, let us see this formula in action with a concrete example. A patient's pancreas secretes insulin at a constant rate of 20 milliunits per liter per minute. The clearance rate constant is 0.5 per minute. We are asked to find the final steady state concentration. Because it is at steady state, the rate of change is exactly 0. We substitute our given values into the equation, setting 0 equals 20 minus 0.5 times C. Solving this gives us a final concentration of 40 milliunits per liter, meaning the patient's blood sugar regulation is perfectly stable. Now that we have used this concept to find a patient's insulin level, let us formally derive the steady-state formula. We start with our differential equation for the rate of change. To find the steady-state concentration, we must recognize that steady state means the hormone level is no longer changing over time. Therefore, we set the rate of change, dC over dt, to exactly 0. By setting the left side to 0, we are mathematically stating that the amount of hormone entering the blood perfectly equals the amount leaving the blood. Continuing from our balanced equation, we can now isolate the concentration. We rearrange the equation by adding the clearance term to both sides, giving us K times C equals S. This simple algebraic step represents a profound biological truth. The rate of clearance exactly equals the rate of secretion. Finally, dividing both sides by the clearance constant, K, gives us our direct formula. The steady-state concentration C equals S divided by K. This shows that final concentration is directly proportional to secretion and inversely proportional to clearance. So we have proven how the body maintains a steady concentration of hormones in the blood. This brings us to the next crucial step. What happens when the hormone reaches its destination? Once a hormone reaches a target cell, its physiological effect depends entirely on how many receptors it can successfully bind to on the cell surface. Even a tiny amount of a highly potent hormone can trigger a massive response. Understanding this receptor binding is essential for determining the correct dosage of endocrine medications to ensure they are both safe and effective. To quantify this cellular response, we use a standard binding equation derived from chemical kinetics. The formula states that B, the number of bound receptors, equals Bmax times the free hormone concentration H, divided by Kd plus the hormone concentration H. Bmax is the absolute limit of the cell's response. Kd is the dissociation constant, telling us how tightly the hormone binds. If the hormone concentration increases, more receptors become bound. A lower Kd means the hormone binds very strongly, requiring less hormone to achieve a massive effect. Let us apply this binding equation to a real-world medical scenario. An endocrinologist administers a synthetic thyroid hormone. The target cells have a maximum of 1,000 receptors. The dissociation constant is 4 nanomolar, and the blood hormone concentration is 12 nanomolar. We are asked to find how many receptors are activated. We substitute our values into the equation, 1,000 times 12 divided by 4 plus 12. This simplifies to 12,000 divided by 16. The final answer is 750 activated receptors, meaning the cells are operating at 75% capacity. We have covered a lot of ground today, so let us bring it all together. First, hormonal regulation relies on negative feedback loops to maintain perfect internal balance. Second, the steady-state concentration of a hormone is a mathematical balance between its secretion rate and its clearance rate. Finally, the ultimate physiological effect is determined by how many cellular receptors are activated, which we can calculate using the binding equation. Mastering these concepts is the absolute key to understanding how the human body coordinates its most complex functions.